Welcome everyone to Lab 20, the topic of which is multi-test systems for enteric bacteria. Enteric bacteria are defined as bacteria that typically reside in the digestive tract, specifically in the intestines. There are a wide variety of enteric bacteria, and some of these enteric bacteria are pathogenic to humans, including Shigella, Salmonella, some strains of E. coli, and others. In this lab, you are going to be exploring a tool called a multitest systems, which allows for rapid identification of enteric bacterial species. There are a variety of multitest systems available in commercial laboratories, but the one that we are going to be using in this lab is specifically called the enterotube 2, or the enteropluri test. What this system essentially is a number of different types of selective and differential media that are arranged into 12 different chambers inside this neat little compact tube, which allows you to inoculate all of the 12 chambers at the same time with the same sample of unknown bacterium. And in doing so, you can then look at the results of 12 different tests at the same time and determine the identity of the microorganism based upon the results of those tests. So I'll talk about what those tests are in just a second, but the way that this tube is inoculated is if you look at the two ends here where I have removed the screw caps, there's a blue screw cap on one end on the left and a white screw cap on the right. When you take the blue screw cap off, it reveals a handle that is associated with a needle. And that needle runs all the way down the middle of this tube, piercing through every single chamber of selective and differential media. On the right hand, where we remove the white screw cap, there is the tip of a needle. And this tip will be inoculated with bacteria, and then that bacteria ends up getting dragged through every single chamber of the tube to inoculate all 12 in one fell swoop. So the types of media that are located in each of these chambers will produce different reactions depending upon the species of bacterium that is used to inoculate them. So let's talk about what's going on in each of these different chambers. The first chamber, starting on the left-hand side and moving toward the right, the first chamber is called the glucose/gas chamber. And what this tests for is the bacterium's ability to ferment glucose sugar. A negative result will look like red media in color because there's a pH indicator in there, just like in our fermentation tests. If it remains red, then that means that the organism has not fermented the sugar into acid and dropped the pH to change the chamber's color. If there has been fermentation that takes place, then what you expect is the media will change color to yellow, and possibly a gas bubble will be produced along with that color change. And so there are actually two different components to this first chamber. You're checking to see if there is a color change, and you are also checking to see if there is a gas bubble. The second chamber here is called the lysine chamber, and this chamber contains media that is infused with lysine. It also contains a pH indicator, which will tell you if the lysine is being broken down by the bacteria. A negative result, which you can see in this uninoculated clear tube, is yellow in color. But if it's a positive result and the lysine is being broken down, then it will change to a purplish color. The same goes for the ornithine chamber, which is the third one in line here. This checks for, oh, that's a typo there. It should be the breakdown of the amino acid ornithine. And just like in the lysine chamber, it starts out yellow, and if it changes purple, that counts as a positive biochemical reaction there. The fourth chamber here is called the hydrogen sulfide and indole chamber. And kind of like the glucose gas chamber, this actually looks for two different things. It looks for the production of hydrogen sulfide, indole, or both. The chamber starts out yellow in color, and if hydrogen sulfide is being produced, then there will be a change in color to black. If indole is being produced, then there will be a change in color to red. And if both are being produced, then you would see both black and red in that chamber. Fifth chamber down the line is called the adonitol chamber. This checks for the fermentation of a carbohydrate called adonitol. It starts out as red, and if the organism can indeed ferment adonitol and produce acid, then the pH indicator changes to yellow. This is exactly the same for the next few chambers in the line, which contain the sugars lactose, arabinose, and sorbitol. All of them start out red. If the organism can ferment that sugar or carbohydrate, then it can turn the chamber to a yellow color by dropping the pH. The ninth chamber down the line is called the Voges-Proskauer, abbreviated VP chamber. And what this checks for is the production of a compound called acetone. It starts out yellow in color, and if acetone is present, then it changes to red color. The 10th chamber is called the dulcitol/phenylalanine, which is what PA stands for. Dulcitol/phenylalanine checks for the fermentation of the carbohydrate dulcitol, as well as, at the same time, the breakdown of the amino acid phenylalanine. So this is again a chamber that checks for two things at once. It starts out as green, and if there is a fermentation of dulcitol, then it will change in color to yellow. If there is, on the other hand, a breakdown of the amino acid phenylalanine, then it will change color to brown. Next, we have urea. Urea is a test that we performed previously in the class. This chamber checks for the breakdown of urea. If the urea is indeed broken down, then the yellow color in the chamber is changed to pink as the pH of the environment is increased and it becomes basic. And then last but not least, there is citrate. This determines whether the bacterial species is capable of utilizing citrate as a source of energy. The chamber starts out as green, and if the bacterium is capable of breaking down citrate, then it changes to blue in color. So before we go any further and we take a look at an example tube, let's take a look at Professor Fotini-Sioris doing a demonstration of how this tube gets inoculated.
The materials for the enteroplury test is an enteroplury, a blue cap and a white cap, and the different tests, the glucose test, and they're written in the front, the lysine test, the ornithine test, the hydrogen sulfide indole test, adonitol test, lactose test, arabinose test, sorbitol test, the VP test, the dulcitol phenylalanine, the urea, and the citrate tests. This is what the original uninoculated with no bacteria enteroplury looks like. On the back, you will notice there's tiny little windows that actually have a film over them, and you can pierce a hole through them to allow aerobic conditions. The last four compartments are anaerobic. There are no windows. The bottom, there's film, and this film can be actually removed for the addition of other reagents to run tests. And you will see this in the results. If we unscrew the blue cap end, you will notice a metal that is folded in order for you to be able to handle it on that side. The white cap end has a needle, and this needle you should never touch because you don't want to get it contaminated. So this is an enteroplury test. For materials, we also have unknowns. We have unknown 1. Again, we don't know which bacteria these are. We are going to guess them using the enteroplury, not guess them, identify them. Unknown 2, unknown 3, unknown 4, unknown 5, and unknown 6. Here's the 6 enteroplury that we will use, 1 for each bacterium unknown. So now we will pick up the unknown number 1 and show you the procedure for this enteroplury. The first thing we want to do is label the number 1 somewhere on our enteroplury system, preferably on the top where you can see it. So we put a number 1 because that's our number 1 unknown. And we're going to try to identify what this unknown name is. We unscrew the blue cap, and we also unscrew the white cap. Remember, the one end is a needle, and the other end we can touch with our hand. Here's the unknown one. We uncap it, take the lid off, and we pick up some of the bacteria growing on this unknown one plate, just like that. Usually you can see a little piece of the bacterial culture on the end of the metal, and now with the other end we pull the metal rod through all of the agar compartments, inoculating them, placing bacteria on these agars. We go all the way watching the rod until we reach the last agar compartment, and we inoculate that as well. So we want to make sure we get bacteria into each compartment. And then we have to push the rod back through into the enteroplury device. And sometimes we just have to jiggle this a little bit, and then once we get it to go through, there's a little notch there that you probably can't see. And at that notch, we break off the rest of the needle. Right there. Now, using the sharp edge of this metal piece, you can pierce holes in those little windows we talked about to allow for aerobic conditions. And some people like to dip it in alcohol just to disinfect it before they do this. And we just pierce holes through the film in these little arched windows. And there's about 8 of them. The last 4 do not have those windows, so they will be anaerobic conditions for those tests. No air, no oxygen. Okay, once we are done, make sure we dispose our little metal piece in the hazard trash and cap. The blue cap usually goes on the one end, and that's the end where the glucose is, the glucose test, and then the white cap goes on the other end. We do the same procedure for all bacteria until we have 6 enteroplury systems inoculated.
So that's how the enteropluri tubes get inoculated, and then they are incubated in an incubator at 37 degrees Celsius for 48 hours. And after that takes place, then you will be able to observe the results and determine whether there have been any biochemical reactions taking place in any of these 12 chambers. Most of the chambers can be observed on site to determine if there is a reaction that took place. However, there are 2 chambers in particular that you need to add a chemical compound to in order to be able to see if a reaction has happened. So let's take a look now at another video very quickly of Professor Sioris adding that reagent, that chemical compound, to the 2 appropriate chambers to determine whether a reaction has taken place.
Before we read results, we want to make sure that we burn our loop nice and hot, and we burn the film on the underside of the enteroplury system in 2 compartments. The VP compartment, which is the one that you can see the film is removed here. Again, we use the hot flaming loop in order to burn the film off of the VP compartment because we are going to add a reagent that will help us complete that test. We do the same for the H2S/indole compartment, hydrogen sulfide/indole compartment. We burn a nice big hole where we can drop some reagent drops and complete that test. And again, we only do this with two compartments, the VP and the H2S/indole. You can see here quite a bit of the film has been removed. And now we are ready to add to our VP section solution A, and after we crush the ampulla that's inside, and therefore we can allow for 2 drops of the reagent A to be dropped in, and then 2 drops of reagent B. There's going to be a chemical reaction within 5 to 10 minutes. We also add the indole reagent to the H2S/indole compartment in order for the chemical reaction to happen there.
So once the incubation has taken place and once the reagents have been added to the 2 chambers indicated in Professor Cioris's video, you can then interpret the results, and the beauty of these tubes is that they allow you to come up with a 5-digit code that represents the identity of that bacterium based upon the biochemical reactions that took place. So in the results, you will be provided with a key that looks like what you see above here. We have a figure showing what counts as a negative result in the enterotube, and just below it, what counts as a positive result. And you'll also be provided with a decoder, which looks like this and allows you to come up with that 5-digit code based upon interpreting what positive results have happened here. The way that this decoder essentially works is if there is a positive result for one of the biochemical tests, then you write the indicated number in the box below here. So in other words, if the test is positive for glucose, you write the number 4 in this box. But if it is negative for gas, for example, then you write the number 0 in this box. After running through all of the different things that the enteropluri tube tests for, then you sum up the sets of numbers as indicated by the brackets here and come up with a 5-digit code that is unique to that species of bacterium. So I know this is a little bit confusing, and therefore we are going to walk through the decoding of unknown number 1 together, and then you will be responsible for decoding unknown 2 through 6 on your own. So let's take a look at unknown number 1. This is the result that we get up at the top here after the incubation has taken place and after the reagents have been added to the 2 chambers. We're going to go through each of the different tests and chambers and determine whether or not we should add a number to the decoder. So here's our example of what a positive versus negative result looks like. And as we can see, in the very first chamber here, the glucose and gas test, we have started out as red, and a positive result counts as yellow. Our glucose chamber is definitely yellow, and therefore we want to put the number 4 in that very first spot there. There is, however, not a gas bubble in this chamber. And the gas bubble can be difficult to see if you're just looking at a static image like you're going to be provided. And so you will have labeled for you whether or not a gas bubble is present so you know definitively one way or another. But there is no gas bubble in this chamber. You'll have to take my word on that. Therefore, we add a 0 to the second box. Lysine is the second chamber. We know that if lysine had a positive result, then it would be purple. And so this is not a positive result for lysine. We add a 0 to that box. Same goes for ornithine. Ornithine is the third chamber. Started out as yellow. If it was positive, it would be purple, but it has stayed yellow. So ornithine is also a 0. Hydrogen sulfide and indole started off as yellow. And if it was positive for hydrogen sulfide, it would be black. If it was positive for indole, it would be red. It is neither. Therefore, both of those boxes, hydrogen sulfide as well as indole, get a 0. The next 4 chambers are relatively easy. The carbohydrates and sugars, adonitol, lactose, arabinose, and sorbitol, all are based upon the same fermentation principles. They started out as red. If any were positive, they would have changed to yellow. None of them have. So therefore, we have 0s in all 4 of those boxes. Vogue's Proskauer started out as yellow. It would be red if it were positive, but it is not, also a 0. Dulcitol and phenylalanine is the next chamber. And there's 2 alternative results that you can get here. Either the green turns to yellow or the green turns to brown. In this case, the green has turned to brown. So while we are negative for dulcitol fermentation, we are positive for phenylalanine breakdown. So we add a 0 to the dulcitol and a 4 to the phenylalanine. Next we have urea. We can see that urea started out as yellow. If it was positive, it would be pink. And indeed, it has turned to pink. So we write a 2 in that final box for urea. And then lastly, citrate starts out as green. If it's positive, it turns blue, and it has turned blue. So therefore, we add the number 1 to that last box. Now we sum up these numbers, as indicated by the brackets, to come up with our 5-digit code. Our first one is 4. Our next 3 are very simple, 0, 0, and 0. And our final one is 7. So the 5-digit code that corresponds to this bacterium is 40007. Now what we get to do is take this 5-digit code and turn to a code book that includes all of the possible 5-digit codes that you can get out of this test so that we can find the species of bacterium that corresponds to the code. Here is what the codebook looks like. This is provided to you as a PDF on Blackboard that you can download. And the quickest and easiest way to go about this is to use your Control F function. By hitting Control F, you can then type in the number of your unknown in the Control F box. And then you can use that to find the code in this very long code book, because this is actually over 200 pages long and you don't want to go through all of the pages manually to find your code. So use Control F. And when we finally locate our code in this code book, 40007, we can see that there are actually 3 different possible enteric bacteria that this can be. We've got Providencia stewartii, Proteus vulgaris, and Proteus mirabilis. And the one that we inoculated was Proteus vulgaris. However, for the purposes of answering a question in your lab assignment, you would want to write all 3 because there's no way for you to know definitively which one of these it is. So if there's 1 bacterium for the code, put down the 1. If there are multiple possibilities, put down all different possibilities. And that's how you will go about decoding the other 5 bacteria in this set.